Hi, good afternoon, everyone. This is George Yohrling from HTSA, and Happy New Year, and welcome to our first installment for 2018 of the HTSA Research Webinar Series. I'm really excited to be joined with my colleague here, Leora Fox, who is the Manager of Research and Mission Programs, and a recent addition to HTSA, who will be joining and presenting with me in hope of giving you a 2017 HT Research Year in Review. Before I get started, there may be some new folks to the HTSA webinar program, and just to let you know that on the right-hand side of your screen in the menu, you can click on the question section and type a question for Leora or myself at any point during this webinar. And at the end, we hope to leave plenty of time to answer your questions. Just to let you know, just in case you have to dial off or you know some folks that may benefit from hearing this information, we are recording this and we'll be archiving it on our website. In particular, probably the best way to view it within a week or so is to, if you get your binoculars, you can kind of zoom in on these small icons on the top right and click on the YouTube icon in the middle. Take you to our HTSA YouTube channel, where all of these HTSA research webinars, as well as other interesting caregiver corners and other videos, are stored and can be viewed at your convenience. Just to give you an update, after this webinar, we have 3 exciting webinars planned for the rest of the winter. In February 7th, Wave Life Sciences will be presenting on developing potential treatments for selectively lowering mutant Huntingtin. That's February 7th. On March 21st, we'll be hearing from Dr. Edith Pfister at UMass Medical School in Worcester, where she's going to talk about how sheep can help us develop Huntingtin lowering therapies. This is using using sheep to look at the delivery of Huntingtin lowering viruses expressing Huntingtin lowering drugs in large brains such as sheep. And finally, on April 18th, Dr. Jack Riedling from the University of California at Irvine is going to be presenting the results of his exciting new paper coming out of their lab on neural stem cells as a therapeutic candidate for HD. So this is a slide that describes the mission of HTSA, and the mission is to improve the lives of everyone affected by Huntington's disease. And at the center of that mission is research. But one of the other things that's very important, and we're going to talk a lot about the research that HTSA has funded, as well as research that's going on around the world in the past year. But at the center, another important aspect is clinical care in our Centers of Excellence program. And I just wanted to, since these are places where not only do patients and families receive multidisciplinary care for their HD, but they also are places where families and patients can go and participate in meaningful HD clinical research. And we're committed at HTSA to expanding this network of expert centers around the country so that it decreases the geographical, financial, and other burdens to families to get to these places. And I'm happy to report at the end of 2017, we have 41 centers of excellence around the country, plus 5 additional collaborating clinics. These are clinics or satellite clinics that are collaborating with our larger level 1 centers of excellence. That brings us to a total of 46 different clinics around the country where patients can go and expect expert care for their HD. Since we rebooted this program, the budget committed towards it is nearly $1.2 million since 2015. HTSA, one of our ambitions is to ensure that the pipeline of researchers and clinicians is primed and ready for whatever the future may hold for Huntington's disease. And so through our research programs, we really want to provide the necessary experiences and resources to young scientists and clinicians to get them a head start and hoping that they will choose HD as a career. One of our exciting programs for undergraduates and medical students is called our Donald King Summer Research Fellowship. And this year we awarded 4 fellowships. This is the most we've ever awarded. And here you can see 2 of the recipients, Kiki Kim from Columbia University, who's working with Ai Yamamoto, basically looking at a project to understand how brain cells, quote unquote, take out the trash, and that trash being mutant Huntingtin in Huntington's disease. We also funded Christopher Gannick at the University of Central Florida, who's working with Amber Southwell. Amber's also an HDSA Human Biology Fellow, and Chris and Amber are working together to develop tests to see how if they can accurately measure mutant Huntingtin in CSF, which stands for cerebral spinal fluid. That's the fluid that kind of bathes the brain. And we'll talk a little bit about that later on when we talk about some of the Huntingtin lowering approaches. Paul Elizad from Catholic University, working with John Choi, was funded over the summer to answer the question, why do HD neurons become damaged and die? This is a long-standing question in the Huntington's field, is why is there a selective vulnerability of the striatal and basal ganglion neurons in the brain over other regions of the brain? And finally, Teal Jenkins, who's a medical student at University of Washington Medical School, was working with Dr. Jonathan Fox, who's actually a professor at the University of Wyoming, and looking to answer a question of how do other infections potentially influence HD? So not only does we know that the Huntingtin mutation causes the disease, but there are other factors, potentially infections, potentially environmental factors that could all play a role in accelerating or deaccelerating the onset of HD. Our largest and our flagship program for HD research is the Human Biology Project, and that really continued to move. We launched it in 2013, and it's moving strong in 2017. We awarded four grants to 4 outstanding researchers from around the world. One is Dr. Marina Papuzzi at the University College London, working with Dr. Sarah Tabrizi. And she's trying to answer the question of what factors influence our cognitive or thinking capacity in HD. Dr. Marta Biagioli is an assistant professor at the University of Trento in Italy, where she's looking to understand if we can use this novel, what we call a circular RNA, as a blood-borne HD biomarker. Dr. Ali Koshnan, who's a scientist at California Institute of Technology in Pasadena, was funded to look at what are the effects of the Huntingtin gene in the gut. And finally, Andrea Rutenek is a graduate student at the University of Miami Medical School, and she's working with Antonio Barentios on a program or on a proposal to see if she can harness metabolic pathways, endogenous metabolic pathways, to protect people against the harmful effects of the Huntingtin protein. So in all, this program has awarded upwards of $3.6 million to really outstanding clinicians and scientists from around the world. We look forward to hearing from them and others at the HDSA convention and webinars just like this. Last year, HDSA launched a new research program called the Berman Topper Family Fellowship, where we awarded one outstanding young fellow 3 years of funding to really get their feet on the ground and provide them the necessary resources to answer some very important scientific questions related to HD, but also to give them a long exposure to the HD community, and hopefully that they'll continue with this course of research as they become independent in their career. And this year, we're really excited to be able to award 2 of these fellowships to 2 outstanding young scientists. One, Dr. Tamara Mayuri, who works with Dr. Ray Truant at McMaster University. And Tam is working on a project to see if we can find drugs to boost DNA repair and delay the onset of HD symptoms. And DNA repair is something we'll touch on in a little bit, because there's a lot of smoke around DNA repair pathways and their potential role in the etiology or the biology of Huntington's disease. And finally, we also awarded a Berman Topper Family Fellowship to Dr. Sarah Hernandez from University of California, Irvine, who's working with Leslie Thompson to investigate or to answer the question, does development of and maintenance of the brain cell structural supports play a role in Huntington's disease? In addition to the research programs that we fund at HDSA over the past year, we have a lot of other opportunities and things, forums that HDSA has done to try to make people aware of all the great things that are happening in HD science. Throughout the year, we have education days. Hopefully, many of you have been to some in your local area, where we host between 40 and 50 of these education days, whether in collaboration with our HDSA chapters or centers of excellence. In addition, we host the Saturday of our annual convention, which this year will be taking place in Los Angeles. We hope you can join us for that. But last year it took place in Schaumburg, Illinois, in June. We had a fantastic research forum where we get all the latest updates from exciting science going on. We continue and these HDSA research webinar series and hope to host one at least once per month. HDSA has remained a contributor and supporter of HD Buzz. HD Buzz is a hugely important asset to the community because they take very difficult scientific content and are able to distill it down into easily understandable terms and for the families. Each year we craft a annual research report, and that's shown here in the lower right. This is the cover, and hopefully this was just released, I believe, last week online. So it's online, hdsa.org, so please take a look at it. It's going to give you a little bit more detail about the things that we're talking about today, as well as some other things that are going on that we just don't have time to cover during the course of this webinar. And finally, as I alluded to, HD research has, our programs have grown to a point, and the pace of HD research has accelerated to a point that in order to keep up with all the things going on, we've made a strategic decision to expand our program or our staff here. We've added a new staff scientist, Dr. Leora Fox, who comes to us actually from Yamamoto's lab at Columbia University, where she worked on Huntington's disease for many years. And you'll be hearing from her in just a few minutes. HDSA was, in 2017, HDSA was very excited to join, partner with the two other nonprofit HD or patient-facing organizations, the European Huntington's Association, as well as the Huntington Society of Canada. And together, the three organizations formed a coalition that we're calling HD Cove. HD Cove stands for the Global HD Coalition on Patient Engagement. And patient engagement is becoming critical and a very important thing for pharmaceutical and biotech companies to consider before they go to the FDA or other regulatory agencies to get their drugs or trials approved. It's really important that the patients and caregivers of Huntington's disease families and patients provide the meaningful patient experience or perspective to companies so that the trials meet the needs of those families. So HDSA, EHA, and HSC are all proud members of the advisory. We all represent the advisory board, and we're excited to bring this global team of patients and volunteers together. And this team will be working in 2018 very closely with many of the major pharmaceutical companies that are working in the HD space. There was a lot of really exciting science, I think, that happened in 2017, and I think Leora is going to talk about some of the most exciting stuff in terms of Huntingtin lowering, but I want to reiterate that there's been a lot of really exciting basic research headlines in 2017, and we sat down and tried to think of what were three of some of the more exciting things in our mind, and one was a recent paper from Lauren Byrne and Ed Wild, group at University College of London, looking at neurofilament light chain, or NFL, as a potential blood-borne biomarker for HD. Another was some work being done to explain the variation in the age of onset of Huntington's disease, why people with certain CAGs get the disease at a certain age versus those with the same CAG length get it at a much later date. And finally, a word that, or an acronym that many of you have probably heard in the news or in papers called CRISPR. And there have been a few papers that have shown that CRISPR, this technology, which we'll talk about in a minute, was successful in doing some gene editing in HD mice models. This was the neurofilament light chain, or NFL, was identified as a potential blood biomarker, as I mentioned, in a paper that came out from Lauren Byrne and Ed Wild, and was actually the subject of a recent research webinar, which you can find on our YouTube channel if you were interested in hearing more details about this exciting work. But basically, what we see, this is using Track HD data, an observational study of a few hundred HD patients from around the world. And you can see this is the concentration of NFL, neurofilament light chain, in plasma of HD patients, and that the controls have a low level. And with increasing stages of the disease from early pre-manifest all the way to stage 2 manifest HD, you see a increase in the levels of NFL. And likewise, that increase in NFL concentration is seen as the CAG repeats increase. So this is a very interesting work and could be useful, very useful in clinical studies in the future. Question that we get a lot, and it's a really interesting question we'd love to find out the answers to this, is to explain the variation in the age of onset of HD patients. So let's think about looking at these green and red dots on the screen on the right-hand side. This is a graph that many of you have probably seen over the years. On the left-hand side, it says age of onset. This is the age of neurologic onset, typically characterized as motoric or motor onset. And on the x-axis, or on the bottom, you'll see expanded CAG. And this is your CAG repeat length within the Huntingtin gene. And what we see is that there's an inverse correlation between the CAG length and the age of onset, meaning as the CAG expands, the age of onset gets younger and younger. But in the case, you can tell that there's a lot of noise here in these data, and taking into account these folks here in red that have 40 CAGs, and they had a neurologic age of onset at age 40, yet these two in green had a neurologic age of onset at 70 with the same CAG repeat, a 30-year difference. And that would be pretty remarkable if we could figure out what's causing that, and are there genes at play, and potentially design drugs that could recapitulate what we're seeing here on the screen between the red and the green dots. So some really exciting work. A few years ago, there was a study that came out, and it was called a genome-wide association study. This was looking at thousands and thousands of HD patients, looking at their genome, and they were able to find some regions within different chromosomes that were suggestive of some genes that could be playing a role in explaining this variation in the age of onset. These would be called genetic modifiers. In 2017, a paper from Davina Hensman-Moss and Leslie Jones that appeared in Lancet Neurology, and there were others, but they highlighted, focused on a region in chromosome 5 where there was some noise from the original paper, the original GWAS paper, and they found that a gene called MSH3 there was a mutation in this gene that showed that when it was mutated, there's a decrease in the progression in the UHDRS score, suggestive that we could potentially, if you could potentially modulate or inhibit that gene, you could potentially have a beneficial impact on the course of Huntington's disease. And what we've known, this gene is not some unknown to many HD researchers. We've known about this gene for many years. Work out of Vanessa Wheeler's lab in Mass General has shown that these genes, this MSH3 gene and other related genes, when you knock them out or shut them down genetically, we can do that in mice, we're able to stop a process called somatic instability. And somatic instability is the ability of that CAG track in the Huntingtin gene to expand. And we know that that gene expands over time. It's very unstable. And by inhibiting or knocking out this gene, MSH3, we're able to inhibit that process. And finally, there's been a lot of press around CRISPR. It's the acronym, I'm going to see if I can get this right, is, because I know you're all wondering what it stands for, and there'll be a test after this. I'm going to ask Louise if she's paying attention. Clustered, regularly interspaced, short palindromic repeats. It's a mouthful, and you can see why that's why we call it CRISPR. But basically, CRISPR is this cool kind of mechanism that was identified in bacteria, and bacteria adapted this mechanism to protect themselves against viral infection. And they can protect themselves by changing their genome so that they don't die from viral infection. So scientists are now using CRISPR, which will involve this Cas9, which is a CRISPR-associated protein, Cas9 enzyme, as well as this piece of DNA. It's a guide strand RNA, it's single-stranded RNA. This is the tool. This would eventually, potentially in the future be a drug. But scientist Xiaozheng Li from Emory University published a paper, as did a few others in the field, that showed that they're able to use CRISPR as a tool in the lab to genetically modify HD mice. In the case of Xiaozheng Li's paper, they were able to knock out Huntingtin using this CRISPR technology and restore a lot of function to an HD mouse model. So before I turn it over to Leora for her clinical trial update, there's some really exciting news in the clinic that happened. It seems like so long ago, but it really wasn't. In April of 2017, the HD community was happy to see that we had a second drug approved for the treatment of chorea associated with Huntington's disease. And this drug is marketed by Teva Pharmaceuticals and is now referred to as Austedo. So this is an oral drug that controls chorea, the uncontrolled movements of Huntington's disease. It's very similar to kind of an analog or derivative of the xenazine or tetrabenazine, which was approved a few years ago to treat chorea. This is the result of a study called the First HD trial, where this study met its primary endpoints, where they showed that the drug was able to decrease the total motor score and improve the total motor score and total maximal chorea score of HD patients. In addition, it has fewer side effects. They found it had fewer side effects than tetrabenazine, and in addition, it could be dosed less frequently. And the pipeline, before Leora gets into the pipeline, as you see, it really starts with this global observational study called Enroll HD, which is moving strong in 2017. And there are a few changes that were announced and will be implemented as we move forward in 2018 to this program. As mentioned, it's a global observational study. It's open to HD families and patients around the world, with nearly 200 clinical sites available for families, with 3 goals. Goal is to help us understand HD in people, very similar to the HD Human Biology Project, by monitoring the symptoms as they appear over time. So it's a longitudinal study, tracking people over time. It will help us improve the design of clinical studies to give us clear answers more quickly, and will also help us recruit, hopefully, for studies more quickly. And finally, it's helped to, we hope that by tracking thousands of patients across many countries over many years, we'll identify best clinical practices for HD care and hopefully incorporate that into the clinic in the future. So this is a study that's been going on for a few years, and what we'll start seeing is a change as we move to an era of studies that are looking to that lower Huntingtin, potentially be disease modifying and delay the onset or the progression of Huntington's disease. We're going to need more at-risk and pre-manifest and early stage HD patients to participate, because those will be the candidates for these future Huntingtin lowering studies. And so there'll be an increased push for recruitment of those individuals. There'll be a transition. This was announced by Jamie Levy and folks at the HTSA convention in June, that they'll be transitioning current participants that are in the more advanced stages of HD. They'll still be able to participate, and it's important that they participate, but they'll be transitioned to a less burdensome or an easier protocol, which they're going to be calling Enroll Light. And finally, in the future, hope to utilize in Enroll mobile health applications to accommodate younger working and remote participants in something tentatively called self-enroll. So stay tuned for that. So now I'm going to turn it over to Leora, who's going to talk to you about the latest progress in the HD pipeline in 2017.
Hi, everyone. This is Leora, and as George said, I'm going to take the baton now. I'll be talking about drugs that are currently in the clinical pipeline, and I want to mention that we are currently aware of more than 20 companies that are working on drugs for HD, and there is more and more industry involvement because of great platform studies, big observational studies like Enroll, and because of a really amazing and engaged community. So thanks to all of you for your interest in research, and thanks again for listening in today. So I'm going to focus on trials that we had either some news about in 2017, like PRIDE and Gotto, trials that are currently recruiting right now, like Signal and STARE. And then I'll talk about Huntingtin lowering, like the Ionis Roche trial that has been all over the news recently. And I'll mention another set of Huntingtin lowering trials by a company called Wave that is also coming soon to the clinic. So to begin with trials that we got news about this year, I want to start by talking about PRIDE HD, which was a phase 2 trial run by Teva Pharmaceuticals for a drug called pridopine. It's taken by mouse, and it's designed to stabilize dopamine, which is a neurotransmitter or a brain chemical that helps to control movement and mood. So this year we learned that the study found that when this drug was taken for 26 weeks, it didn't improve total motor score, but it did improve total functional capacity, which is basically a measure of how people function in their daily lives. And you can see on the right how that's measured in terms of how people are functioning at work and at home and with their finances, and pridopine in this trial helps to improve that overall. So this finding, in addition also to some new data on the biology of how the drug is working in brain cells, has led to the planning of the next phase of this trial. And it's not currently recruiting, but we are keeping a close eye on that information, and we'll share if we hear more about the next steps. So next I want to talk about another phase 2 trial called Gotto HD, which involves another drug from Teva Pharmaceuticals called laquinimod, which is also an oral medication that's in the study was taken for 1 year, and it targets inflammation in the brain. And the main goal of this study is to see what the effect of laquinimod is on movements in HD, but also looking a bit at other symptoms like changes in emotions and memory. The study was delayed for a little bit because of some heart-related side effects at higher doses, but those issues got resolved, and recruitment is complete as of this year. We learned in 2017 that the final clinic visits are happening in the middle of 2018. So we'll expect results of this study in the fall of the coming year, and from there they'll determine whether the drug can move on to the next step of clinical testing. So now I'm going to talk a little bit about 2 trials that are currently recruiting. This one's called the STARE study, and it's from Azavan Pharmaceuticals, testing a drug called SRX-246. And this is also an oral medication that's taken twice daily during the study, which lasts 12 weeks. And the drug targets a receptor called vasopressin 1AR, and this plays a role basically in sending chemical messages in the brain that can sometimes cause anxiety and aggression. So this studies specifically looking at these types of emotional symptoms in HD. And they're trying to recruit 108 patients for this study. It's ongoing at 22 different sites all over the U.S. At the end of this presentation, I'll talk about HD Trial Finder, which is a great way to learn more about this trial and other studies that are currently recruiting. And the next study I'll talk about is the Signal trial, and this is for a drug called VX-15 that is made by Vaccinax. And this drug is taken monthly through an IV. It can last for about 12 to 18 months, and it's an antibody that blocks another receiver of chemical messages in the brain called SEMA4D. And the goal of the study is to understand whether the drug can decrease inflammation in the brain and possibly increase the health of nerve cells. So we hosted a webinar in September, actually, about the early results of this trial. And they did some imaging, like MRIs, and they showed results that VX-15 does affect the volume of the brain. So if you're interested in more details about this, you can watch the webinar on our YouTube channel, or you can go to hdsa.org and look for the webinars, and you can also read more about it at HD Trial Finder. Recruitment is also ongoing all over the U.S. So before I move on to talk about Huntingtin lowering, I want to talk about some data that came out very recently from the FDA about overall drug approvals in 2017. So in the top graph, you can see how many drugs were approved per year over the last 10 years. And 2017 was actually a great year. In addition to these 46 shown here, there were also 6 in another category of recombinant therapies, like genetic therapies. So a total of 52, and this is actually the best year since 1996 when there were 53. And you can see that there's a general trend. We had a little blip last year, but over the last 10 years, there's basically been an increase in the number of drugs that the FDA has approved. And I want to point out that 39% of these new drugs in 2017 were for rare diseases with orphan status like HD. So Austedo falls under that category. 78% of these drugs were approved in the US before any other country. So it's really encouraging that more and more companies in the US are interested in looking into treatments for rare diseases, and especially that they are having success with that. So the good thing about fighting a disease like HD is that we know what causes it, and we know exactly what the genetic error is, and Huntingtin lowering drugs have a lot of promise, in part because they're designed with the actual cause of HD in mind. So most of you are familiar with the idea that HD is caused by a mutation in the DNA within a gene called Huntingtin. And DNA is made up of nucleotides, which are these building blocks that we represent with the letters A, G, C, and T. So everyone's got this gene called Huntingtin. There's a sequence of letters in it, CAG, and in most people that CAG sequence does get repeated a few times. But in a person with HD, that CAG gets repeated over and over, more than 36 times. And that DNA error gets passed on into an RNA message. And this RNA message then gets turned into this very long and unruly protein that can't really fold up properly or do its job properly. And over many years, that protein can build up and become harmful. So we believe that if we can interfere with one of these steps, there will be eventually less of that harmful protein, and that could hopefully improve symptoms or delay the onset of symptoms. So to use a really simple analogy for this, we can think of the DNA sort of like a cookbook full of recipes, and say you want to make cupcakes. So you take your cookbook and you write down a copy of that recipe, and then you bake that recipe into cupcakes. But imagine that there is now a mistake in that recipe, like the flour is missing. So now you've got most of the right ingredients, but your cupcakes don't really come out like cupcakes are supposed to. Well, we want to find a way to make less of those bad cupcakes. So you could potentially correct or remove the recipe from the cookbook itself. You could stop making copies of that recipe, just stop using that recipe, or you could make the cupcakes and then throw out the cupcakes. And this is sort of an analogy for how Huntingtin lowering therapy works by addressing that mistake at the DNA, the RNA, or the Huntingtin protein level. And there are researchers and companies working on this problem at all of those levels. And the drugs in clinical trials right now are called Antisense oligonucleotides, or ASOs. And these ASOs attack the RNA message, basically. So this Huntingtin lowering strategy is sort of like crumpling up a bad cupcake recipe and never making the cupcakes. And so the way these drugs actually work, it's like a tiny string of man-made nucleotides. So the same kind of letters that make up the genetic code, and it sticks to the RNA message for Huntingtin. And then the brain cell goes, hold on, this RNA looks sort of funny. It's got something stuck to it. And the cell sends out its cleanup crew. So then there's less of this RNA message around, and so less of the mutant Huntingtin protein then gets made. And what current trials are working on is testing whether lowering this harmful protein by interfering with this RNA using the ASO, whether that can slow or stop the progression of HD. And so most of you are aware of the breaking news from December 11th, which is that Ionis Pharmaceuticals announced that the safety trial for their Huntingtin lowering ASO was successful. So they tested this drug in a small group of people outside the US, and they found that in this small group, the drug was safe, there were no dangerous side effects, and the drug did in fact lower the amount of Huntingtin protein in people who participated. So this is a really exciting step forward for HD. The whole community is really excited. We're excited over here. Researchers are excited, and it comes with a lot of hope, and it also comes with a lot of news articles and a lot of questions. So I want to focus on the facts. There were 36 people in this trial. They received different doses of the drug once a month for 14 weeks, and afterwards they had regular clinic visits for another 29 weeks to make sure that nothing dangerous was happening for a while afterwards. So the drug is delivered through a needle to the spinal column, which is known as an intrathecal injection or a spinal tap. That sounds a bit scary, but it's actually quite common, and it's the same way, for example, that an epidural is delivered during childbirth. So the drug was shown to be safe, but a really, really important point that we want to convey about the findings of this study so far is that we don't know yet whether the drug had any effect on people's symptoms. So this part of the testing, this phase 1b/2a study, was only to see if it was safe and if it could lower Huntingtin protein, and that was successful. So the next phase of this study is going to be carried out by a large pharmaceutical company called Roche. It's going to be a larger study, probably with a few hundred people, and it will include sites in the USA. We know that. However, we do not know anything about where those sites are, and we're not entirely sure yet what the eligibility will be, who is going to be able to participate. At HGSA, we're going to share any new information that we have about the trial with the community in the coming months through HD Trial Finder, which I'll talk about in a moment, and through our website, through our social media. We're all very, very excited and hopeful, and we know that Roche is working very hard to move this forward and to make certain that the trial is designed and carried out in the best possible way, and that involves very careful planning of the next stage of the trial. So that is what's currently happening. And so I want to add that HDBuzz.net has some really great recent articles about the trial and answering people's questions about the trial, and we borrowed part of this from them because we think it's really excellent advice from an expert HD doctor and researcher, Ed Wild. So here are some pieces of advice if you're wondering about participation and how to get involved in research studies. So the first piece of advice is to make sure that you are being regularly seen at an HD clinic that has a research interest. such as an HDSA Center of Excellence or another site that would be able to refer volunteers to other clinical trial sites. The second piece of advice is to sign up for Enroll HD. It is a really great platform. It's used as a recruitment database for clinical trials. And when teams are looking for people who are interested in participating in research and have participated in research, and there is information about the course of their disease and participation, this is the first place that people will look. So if I can stress anything, it's that Enroll HD is a great thing to be involved in if you're interested in research participation. The third piece of advice is to take care of yourself because it's going to take some time to start the next trials. It takes a long time to make sure that clinical trial sites are ready, and the healthier that you are, the better the chance of being in the next trial if that's something that you want to do. So keeping up with doctor's visits, different therapy visits, staying active and exercising and eating properly, just all of the regular advice for taking good care of yourself and continuing to seek advice if anything changes with your symptoms. And the fourth piece of advice that we want to add is to sign up to get alerts from HD Buzz and sign up for HD Trial Finder. And both of these sites are great sources of information and a way to directly match with clinical research. So I want to just briefly mention another ASO study that's also going to be entering an early safety trial in the US, and that's through Wave Life Sciences. So this is actually a similar therapeutic approach to lower Huntingtin protein. So since people inherit 2 copies of the Huntingtin gene, one from each parent, most people with HD have one unaffected copy of the gene and one with the HD mutation. And Wave's ASO is specifically designed to lower the harmful form of Huntingtin and leave the normal kind intact. And in this graph, you can see that they tested this in cells grown in a dish, and the drug is doing what it's supposed to do and only lowering the harmful Huntingtin, which is the blue bar. So this drug is also going to be delivered through a spinal tap. And Wave is actually going to be speaking about this upcoming study in our February research webinar, as George mentioned. So we will all hear more from them about the details then. I wanted to highlight one more Huntingtin lowering strategy in development, and this one also focuses on Huntingtin RNA, but it uses a different approach. So this drug is basically packaged inside of a harmless virus called an adeno-associated virus, or an AAV. And the idea would be to deliver that virus into the brain once with a single injection, and then that virus would be able to continue its work inside the brain by creating an interfering RNA, or an RNAi, that would attack the mutant Huntingtin RNA. And the cool thing about this approach is that it would be a single intervention. It could be delivered to a very specific area of the brain and could lower Huntingtin potentially for a long time. But those same features could also be negatives, because once it's in there, it would be sort of a one and done deal that would be difficult to reverse. So this is something that's not ready for the clinic yet, but that is being worked on by several companies, including one called Yuna Cure, and they are going to have representatives at this year's HDSA convention in California. And we're also going to have a webinar in March from a researcher at the University of Massachusetts who's working with a sheep model to explore how to get this type of therapy to the brain. So as I mentioned, there are more than 20 companies working on drugs for HD. This is that full pipeline of everything from early development through currently approved drugs. And in yellow and green here are the ones that I just talked about in a little bit more depth. But I want to reiterate again that the awareness and the willingness to participate in research within the HD community is really incredible, and that's what's helping to move studies along quickly and often faster than in many other rare disease fields. And there's been a really huge outpouring of interest in participating in research in the last few weeks. So I want to take the last couple minutes to focus on a resource that we provide that can help people in the community learn more about clinical trials and get involved. and that is HD Trial Finder. You can find it at hdtrialfinder.org. It's a resource available to anyone in the HD community where you can view an up-to-date database of all of the clinical trials going on in North America, large and small, everything from the big drug trials that we just talked about to smaller studies all around the country where researchers are looking to understand more about some aspect of HD symptoms or about the brain, or maybe an exercise regimen or a device that could be helpful. There's all sorts of studies that help us learn more about how to treat HD and improve care, and they're all on HD Trial Finder. So you can create a profile for yourself or for a loved one there by answering some basic questions. It's all totally privacy protected. The site will then generate a list of studies that you're most likely to be eligible to participate in. And here's an example of what you would see once you matched. All of these personalized trial suggestions to read about, and mousing over the little phone icon will actually give you the contact information for the person who is coordinating the study. And for any help navigating the site, you can call into the call center, which is always listed right on the page, and there are clinical trial navigators that can help you with that. And this year we extended the call center hours to be from 9 a.m. to 6 p.m. Eastern time. There's been a lot of updates in the past couple of months, and we'll continue to update when any trial begins recruiting. So if you haven't matched in a while, I want to encourage you to try rematching, just hitting that button again, updating your info if anything has changed, and checking back. We're also able to send emails to people who have created profiles within a certain distance of a new study. So signing up for Trial Finder is a really great way to stay up to date on research happening near you. There are currently about 3,000 profiles, and this is growing all the time. So with that, I'm going to invite George back in. If he's got anything to say beyond a big thank you for sticking with all of us before we start answering some of your questions.
No, thank you, Leora. That was fantastic. And I'm really nothing to add, but just remind folks that if you do have any questions, we have a few minutes. We can happy to try to answer as many as we can before the top of the hour. There are a few that have come through during the course of the presentation, so we'll try to get through them. One question is coming in regards to the Roche study. Do we know if the new trial will involve a placebo-controlled group? And while the exact protocol has not been identified, it is safe to say that this will more than likely involve a placebo-controlled group. It's just really, we get the anxiety around and the frustration around having some of those placebo groups in some HD studies, but it really is the best and quickest way for drug manufacturers to find out if their drug is working. Another study regarding the Ionis which will become the Roche study is, have investigators or sites been chosen for this study? And the answer is no. The protocol has not been finalized, and nor have sites been chosen. But as soon as they are and they're activated, we'll be in close communication with Roche and their partners, and we'll make sure that that information will all be uploaded to the HD Trial Finder database. So as Leora said, keep checking that as a resource. With a question about access a copy of these slides, as I mentioned, we are recording this, and so this could be will be online on our YouTube channel as a resource. Alternatively, you can email me at HGSA, and I'm happy to try to send you a version of the slides. There's a question regarding genetic testing, and if I could paraphrase it, basically saying, since the availability of the gene test and families are either testing and choosing not to have children, or going through IVF or PGD IVF, or adopting, do we think that this will de-incentivize companies from working on HD? And I think people may have different opinions, but I would say no. I don't think that will de-incentivize folks. HD, the mutation will continue to happen, even if we, the patient population drove down significantly. There are still going to be spontaneous mutations that occur, and we won't get rid of it through that. So there's always going to be a need for effective therapies for HD. And I think just because we're a rare disease, I don't think that would discourage large companies from working on this. And as Leora showed in the FDA data, I mean, companies are really putting a lot of effort on rare and orphan diseases, smaller diseases like HD, because they're really tractable. I think that the companies are learning that, because we know the cause of this disease, of certain mutation in a particular gene called Huntingtin, we know a lot about these patients. This is a very worthwhile and interesting disease to pursue, as opposed to some other diseases, larger diseases that they may be able to make a lot more money, but they're harder nut to crack, such as Alzheimer's or diabetes, where they don't have 100% genetic component or cause to it. Any other questions? Feel free to type them in, but I'm not seeing any more. So if that's the case, I just want to thank you all for joining Leora and myself for this installment of the Year in Review, and we look forward to hosting you all on February 7th and for our next webinar on the selective Huntingtin lowering ASO from Wave Life Sciences.
Absolutely.
Thank you so much, and talk to you soon. Bye-bye.
Bye.